My name is Robert Kelly, and Margaret Buckingham and I would like to congratulate Developmental Cell on their 10th anniversary and thank them for selecting our 2001 paper as a paper pick. Our study demonstrated that the mouse heart tube elongates by addition of progenitor cells from adjacent pharyngeal mesoderm, identifying for the first time a population of cardiac progenitor cells now known as the second heart field. Before describing our findings in a little more detail, I'd like to say a few words about the background to our paper. The complex and critical morphogenetic events associated with heart development have long fascinated embryologists. Two sets of experiments from the 1970s, one carried out in the chick embryo by Maria Victoria de la Cruz and colleagues using iron oxide particle labeling of living chick embryos, and one by Virag and Chalas, who performed a detailed light and electron microscopy study of early mouse heart development, demonstrated that the heart tube elongates by progressive contribution of cells lying outside the heart to the poles of the tube. However, the source of these cells was unknown, and these studies were poorly appreciated by the rapidly growing number of scientists carrying out molecular and phenotypic studies of heart development in the 1990s. In fact, these studies were so poorly appreciated that a general assumption in the field was that all the future regions of the heart were prefigured in the early heart tube. In our paper, we characterized a reporter transgene which showed beta-galactosidase expression in the arterial pole or outflow tract of the embryonic heart and adjacent pharyngeal mesoderm, as can be seen in this left-hand view of a mid-gestation mouse embryo. Unexpectedly, NLAC-Z transcripts were largely restricted to pharyngeal mesoderm, suggesting that beta-galactosidase labeling within the heart was due to perdurance of the enzyme in cells that had shut down transcription. The transgene expression profile was the result of a position effect, suggesting that a gene at the integration site was expressed in arterial pole progenitor cells. Molecular analysis of the insertion site revealed integration upstream of the fibroblast growth factor 10 locus, as shown here. We found that FGF10 was expressed in pharyngeal mesoderm like the transgene, as seen in these transverse sections, strongly implicating this gene as the endogenous target of the regulatory elements trapped in this line. That cells moved from a zone of FGF10 expression in pharyngeal mesoderm into the arterial pole of the heart was confirmed by our colleague Nigel Brown at St George's Hospital in London using dye labeling experiments and embryo culture. Together, these experiments provided the first demonstration that pharyngeal mesoderm contributes progressively to elongation of the cardiac tube during heart looping. Coincident with our paper, two studies were published in Development and Developmental Biology from the groups of Margaret Kirby and Roger Marquardt, identifying using molecular approaches and experimental embryology a similar population of progenitor cells in the avian embryo, which they termed the secondary and anterior heart-forming field, respectively. Together, these three papers strongly suggested a model for heart tube elongation by which progressive addition of progenitor cells from contiguous pharyngeal mesoderm contributes to the rapid growth of the arterial pole of the heart during cardiac looping, as illustrated in green in this cartoon. The term second heart field was introduced in 2005 to define those cells in pharyngeal mesoderm that contribute to the poles of the heart tube after the linear heart tube stage. The intervening decade has strongly supported such a model, and many studies have contributed to dissecting the properties of second heart field progenitor cells in pharyngeal mesoderm. These include the key discovery in 2003 using pre-recombinase tracing, also published in Developmental Cell, that a major part of the heart is derived from progenitor cells expressing the transcription factor islet-1 that contribute not only to the arterial pole of the heart but also to the venous pole. Islet-1 positive cells in the early embryo, fetal heart and differentiated embryonic stem cells identify multipotent cardiovascular progenitor cells that give rise to the endothelial and smooth muscle cells as well as cardiomyocytes. Using retrospective NLAC-Z clonal analysis, a technique developed by Jean-François Nicolas at the Pasteur Institute in Paris, we demonstrated the existence of two myocardial lineages that segregate early in development and give rise to the left ventricle, the first lineage, or the right ventricle and outflow tract, the second lineage, with a contribution of both lineages to the venous pole. This study, Mayak et al., was also published in Developmental Cell in 2004. Following on from the identification of FGF10 as the first endogenous marker of the second heart field, work from many groups has addressed the roles of the major intercellular signaling pathways, including the FGF, BMP, Hedgehog, Wnt and Notch pathways in second heart field development, as summarized in this scheme. These experiments have begun to define a series of complex interactions that collectively describe the niche of second heart field progenitor cells by regulating their key properties of continued proliferation and differentiation delay. Altered second heart field development results in congenital heart defects, a point that was rapidly appreciated after discovery of these cells. Failure to correctly extend the heart tube as a result of impaired addition of second heart field cells results in outflow tract alignment defects and failure of the aorta to make the critical connection with the left ventricle required to separate systemic and pulmonary circulatory systems at birth. And particular subpopulations of the second heart field are highly relevant clinically, and defects in their development make major contributions to conotruncal congenital anomalies. For example, we've shown that future myocardium at the base of the pulmonary trunk is dependent on TBX1, encoding a T-box transcription factor and the major candidate gene in DiGeorge syndrome. I'd like to end with a few comments on the evolutionary implications of our paper. An equivalent population of cells to the second heart field has recently been identified in xenopus and zebrafish, opening up the possibility of using experimental manipulation of the xenopus embryo and forward genetics in zebrafish to identify and functionally characterize new regulators of second heart field development. These observations also highlight the conserved role of pharyngeal mesoderm in heart morphogenesis. In fact, pharyngeal mesoderm contains cells that give rise not only to part of the heart, but also to craniofacial skeletal muscles required for opening and closing the jaw and facial expression. And development of these skeletal muscles depends on genes required in the second heart field, such as TBX1. And this link with craniofacial muscles may reflect assimilation of pharyngeal mesodermal progenitor cells to the heart during vertebrate evolution, as supported by recent studies in the Protochordate intestinalis from the Levine and Christian labs. These results reveal the second heart field to in turn be part of a larger cardiocraniofacial developmental field of importance both in normal development and in the etiology of congenital birth defects.